Hello!
Hi there.
Welcome to YouTube Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
My name is Faz, and in a previous life, I got a PhD in microbiology from Imperial College London, where, among other things, I was making flesh-eating bacteria glow in the dark. And since then, I've worked as a research integrity specialist, and these days, I'm an editor for an academic journal.
Every week, we meet to talk about microbiology, and today is our deep dive week, where we look at each figure in a paper that we chose last week and criticize the scientific findings. This is a journal club, so we encourage people in our audience to leave us questions and comments.
And next week will be our news week, where we survey a bunch of different articles and choose which one we're going to do for our deep dive the week after. So make sure to subscribe to satisfy your microbiology curiosities.
You can follow along with the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
Yeah, and we want to hear from you, so please use the comments or tweet us using the hashtag MicroTWJC hashtag. So, yeah,
Today we were talking about swarming bacteria undergo localized dynamic phase transition to form stress-induced biofilms.
Yeah, this is an interesting one because it aims to look at that transition of bacteria from this swarming phase of where they're kind of still mobile and when they go from that into turning into biofilms, which are somewhat different. So we're going to be talking a lot about that.
For those who may not know biofilms, they're everywhere. You've probably encountered them already in your life. There's these pictures, four different examples of where you could find biofilms, sort of gunky accumulations of bacteria that stick to surfaces.
Yeah, and they're everywhere. So I mean, you probably be most familiar with that grody stuff you find in your teeth in the morning, but they're a nuisance in all sorts of other places, or not even like that. There's variations. So
they're not even like I'm trying to think of where do people really like biofilms. In kombucha, right? That's maybe at the top, right? Like that it's a syntrophic biofilm formed from yeast and bacillus. That's a biofilm that you want around. But often like they do have that texture, I think. They have like some sort of like wet, slimy texture. And the idea is that all the bacteria in there are sort of going down like a cooperative, maybe multicellular sort of like living situation where they stick themselves together with a matrix of some sort.
Yeah.
Sorry, I just want to say, and the reason why they might do that is like for resistance, right? Like it's harder to scrape things away when they're sticky like that. It's harder to have chemicals penetrate deep inside to get some of the cells on the inside. So there's many reasons to take on this form of life. It's a very broad term, biofilm.
Yeah. For sure, and they can be a problem. So understanding how they work can be important for the situations where we're trying to create beneficial biofilms, like or in situations where we really don't want biofilms to happen. So things like, I mean, the one that comes to mind is like ships, where there's constantly marine life just forming on the surfaces of them, or things that anything in the water will get a biofilm at some point. So there's a very important kind of.
Yeah, look at that pipe section.
Oh, yeah.
it's a gross. But that can happen. The insides of these surfaces are not treated in a certain way that inhibits the bacterial growth. Bacteria will stick eventually and find a way to, extract the meager nutrients that are flowing through that pipe for some purpose.
Yeah, and think about like hydroelectric plants or like various other things that need lots of maintenance. So biofilms are important. That's the main thing that we want to get across now.
Totally.
And this paper focuses on Bacillus subtilis, which is the model gram-positive bacteria.
Yeah, which is kind of crazy because I feel like during the time we've been talking about SARS-CoV-2, we've been talking a lot about model organisms, and we talk about animal models, right? Like animals as a model for infection. But this is like a bacteria that's a model for other bacteria. Basically, it's kind of like when you think about how much like people probably know at home E. coli, right? That's like that's a standard name in bacteria. Well, actually, Bacillus subtilis is also like a standard name in bacteria, just from a different lineage, I guess, a gram-positive.
Yeah, and when you grow it on a petri dish, it forms these wild like wrinkles and shapes. It's this the biofilms it forms have this amazing structure to them that
Yeah.
yeah.
And a lot of work has been done on those biofilms to know that they're based like there's this three component operon, TAP A, TAS S, SIP W, and you can see like what they look like. You zoom in on the molecular level. They're on the right, left. Well, I'm actually not sure what
That
direction.
will appear on the right, I think, of the image
Right,
for the for
right. Oh,
viewers.
that's
Yeah.
good. It's logical for me. On the right, right, like you can see like kind of a zoomed in molecular image. Um, and basically, inside of the bacteria, it has to export some of these proteins into the surface, and they mix with exopolysaccharides, so sugar-like molecules, and all of that stuff sticks together, and that's what makes the matrix of the biofilm. And without that, so like for example, like TAS A, that's a mutant of TAS A.
Mm-hmm.
EPS, right on the left-hand side, that's a mutant of EPS. Those things don't make either the protein anchors or the exopolysaccharide. And if you don't make that, you can't make those wavy colonies.
Yeah. So it just looks like just a flat bunch of bacteria that aren't forming any interesting patterns.
Mhm.
Um, And so, yeah, bacteria don't exclusively live on biofilms, so they do swim around. So a lot of bacteria have these things called flagella that allow them to exist in a planktonic form, where they swim around, do their stuff.
Yeah,
But
and I guess
yeah,
that's the cool thing about the model of B subtilis, right? Is that it can live both these ways, right? These are just agar plates that they've shown us, but actually you have to change the agar plates in a certain way to promote one version versus the other. So in these agar plates, like there's a bit less agar, and then what happens is the bacteria just kind of like swarm out. Yeah, actually, they
mm,
call it swarming.
yeah.
Um, yeah, it requires flagella. It also requires surfactant. So
Mm,
these are two, pieces of data from the past. We have on the left-hand side, C and D. That's like wild type versus a mutant with a flagella mutant. And so you can
mm.
see if they don't have the flagella, they are not able to swarm out like that. And then
So,
on the left, oh, go ahead.
oh no, sorry, I was about to say like, oh, so they're just like stuck in that dot. So they don't. So once they can flap around a bit, they can spread out and form that big like flower-like pattern. But when they're not, you just like drop them there and they just sit there, be like,
Yeah, they're,
like
they're
yeah,
still growing, right? That small dot will get denser and denser, and like bacteria will grow on top of each other, because nutrients were still able to diffuse in. But of course,
right,
if they are able to move, they will move because you're getting your own space.
yeah.
And then on the left-hand side, this is another thing that's required for the swarming phenotype, and that's a surfactant. So that open arrow, it's kind of hard to see, but you can see like the edge of something, and it's actually I think it's a protein of some sort that they make, and they secrete into their surroundings, and it makes things lubricated, I guess is the best way to describe
Oh, right. I sort of see it. Kind of like past the little noodly appendages on the like far like right hand, and there's a
That's
little like halo circle with a big like black triangle pointing at it.
yeah,
Yeah, I can see that.
yeah. Yeah, so that halo has to go in front of the swarming colonies. So the newly appendages, those are the bacteria swarming out. But so on the left-hand side, there's two halves. The F
Mm,
side, that's a strain of bacillus that doesn't make the surfactant, and on the G
right.
side, that is a strain that does make it. And so without the surfactant, they can't, I guess, swim over that surface. So there's sort of two dependencies for the swarming phenomena.
So almost like the way that slugs produce that little slime in order to be able to move around, these bacteria produce this surfactant in order to kind of create something that they can move around using.
Totally, I think that would be a really good analogy.
Right. So, yeah, this is where they apply physics to this. So, um, yeah, they talk a lot about this thing called active matter, which is this concept used to describe swarms or entities composed of like large numbers of active agents, each of which can consume energy to move around and exert mechanical forces. So here we're talking about bacteria, but they could be describing other things. So I'm not,
this
yeah,
is this is where like this paper is certainly like a biophysics paper, right?
mm,
Like it sits in this interface between biology and physics. And one thing that we know about physicists is they love to abstract things, right, into their basic components so that they can write mathematical rules potentially, right, that then apply and generalize to other things. That's like the grand plan of physics, right, is to describe
yeah.
everything with equations.
I mean, yeah, I feel like biology wants to do that, but just can't because everything with us just it's too complicated for us at the moment. But I think
Yeah,
that
totally.
But I think that as like kind of things get more advanced, our ability to deal with like massive amounts of data becomes better. I feel like that interface is getting a lot tighter now. Like the way that physics is now being able to explain more complexity and yeah,
Mm-hmm.
so like the same thing that like, happened in the 60s where biologists went to chemistry labs and stole chemists and got them to do work for them. I feel like something similar is happening now with like where people are like creeping into physics labs and going like, grab him, okay, do our work for us now. So
Certainly.
so yeah, this definitely feels like biophysics, where we're finally getting some physicists and their expertise to see what their perspective on biological processes are.
Yeah. And I think what I want to talk about in this particular slide in specific is that abstraction that physicists are using when they think about bacteria moving around.
Yeah.
Like essentially, they can be characterized on this axis. They're how fast do they move and how small are they, and what shape are they, right? Like that's the abstraction here, right? So the lens here is that there's all this physics that's being done, right? Like
Mm.
tons of this is a review I think that I'm pointing to the 2016 paper, a review that says look at all of these different small molecules of different sizes and shapes that move at different speeds. There are physical equations that govern how they move, and the field of physics has been looking at this tons of this. Like you can see
Mm.
some of those things are not even living objects, right? Some of them are just like rods of material that like on one side it's like hydrophobic or hydrophilic, so it like pushes them along, or they move them with electricity. Like these things are very abstract, right? All they care
Mm.
about in this physical field is small objects moving by themselves, and then they try to describe that with equations, and they find, oh, there's these patterns that we're seeing, right? And like maybe these generalize, maybe there are things that occur at this scale of physical, right, of the physical world that we can describe.
Right. Yeah, and so in this case, they're looking through the aggregation of these self-propelled particles using the framework called motility-induced phase separation.
Mm-hmm.
Uh, so that's
So the idea
yeah.
of the phases here are like not dense to dense, right? So that's the phase difference. And you can think about that like I think phase separation might be familiar to people, or phases may be familiar to people when you think about the phases of matter, right?
Mm.
Liquid, gas, solid. So here we have this type of phase separation that happens, but it's happening because all these particles are moving around.
Yeah, this is quite interesting because it's almost like the way they're treating it. I mean, because they're treating this like kind of swarm or like group of as if like they're a material. Like,
Mhm.
for instance, if you're like going to a football match, everyone sat down. That's almost like a solid, and then everybody gets up to move out. Then suddenly people become like a fluid. It feels somewhat like that. I mean,
Except in the football match, people have brains, but like in this mode of life, right? Like the physicists actually, they don't even think about like genetic decision making. I think all
mm,
they're thinking about is two things: one, that you can move, and one, that there's some diffusion abilities that reorient that movement. So that's the example, I guess, on the left-hand side. That's what it's trying to show you, right? That like these clusters occur because as molecules jostle around, right, they're kind of all like reorienting themselves. Those arrows, they move around randomly based on like the currents and eddies of the liquid. Maybe not
mm,
totally randomly, but like somewhat randomly. The Brownian motion, right? The random walk. And as clusters form, you can get the scenario where when one thing decides to leave randomly, because the size and the molecules around have a certain speed, eventually one will join the cluster. And so like these types of like jams occur, right? That are spontaneous and are just arise from these very simple principles.
yeah. I mean, it feels very much like actual traffic jams where one person brakes suddenly, and then but through a couple of decisions, you get this cluster of solid cars that
Totally.
moves, but each car in it can leave whenever, but well, not whenever, but the cars involved in that traffic jam always are changing, but the traffic jam itself is kind of propagating in its own
Yeah.
way.
Yeah, I mean, I'm sure maybe people have heard that advice, or like it's kind of an interesting thought experiment around traffic. Is that if everyone in the traffic jam would just move at a slow speed, right, consistently, it would all go faster. But the problem is all this start stopping, right, is like what's
Yeah.
creating the jam. Yeah.
Yeah, that's why in like some places they have variable speed limits. So when there's a lot of cars on the road, they take it down so that it avoids traffic jams. So I mean, this is a fascinating topic. I mean, Google traffic shock waves. There's a CGP Grey video on it that is amazing.
Okay, before we leave this slide, I just want to touch on the right-hand side of it, and that is
mm,
this is the golden. This is why physicists do this, right? Is because you can make these graphs where you get predictions to form, right? So
mm,
like based on two variables, they have like a PE, which is like a certain number. We'll talk about it a bit more later, right? And then the density of something, that's I think on the y-axis in this case, right? You create these graphs where you can get predictions on with a certain speed and a certain density, how likely are these clusters to form, right? And this is what's all been done before, right? Like this is all in
right.
physics. No one's touch. I mean, some people are doing bacteria. Most people are just doing various shapes that move in this way. They're describing the motion, how it comes to form these things. Yeah, that's what physicists have been doing all this time.
Yeah, I mean, I feel like I should talk about advection. So I don't know if you heard, because when doing this research, I did kind of come across this term of the Peclet number, which is the PE, which is essentially. So let's say you've got like something like an ink blot.
Wait,
When you
wait,
put it
one,
into like a
three,
static bit of water, it expands, like it diffuses. But if you put it into a running river where there's some flow, that flow will affect how it diffuses. So essentially a Peclet number tries to quantify the relationship. So like let's say when you have bacteria that are actively moving, there will be a flow of bacteria that are moving in the same direction, and
mhm.
and some bacteria will be carried along with that. And so the question is like how much of that movement is due to the bacteria just diffusing through Brownian motion or through that like kind of swarm movement. And I feel like that
Yeah. No,
so that's
that's a really good point. It's actually that the B figure on the right hand side, that's sort of describing what you're talking about, right? Like those arrows are all pointing in sort of this little direction, right?
yeah.
And they're essentially forming a vortex, but that vortex is holding all these little particles together.
Yeah, so actually, a lot of those particles don't have any choice in where they move because of all the behavioral, the everything around them. So
Mhm.
it's fascinating, and I know that if there are physicists in the audience, they will be able to criticize me because I'm definitely getting a few things wrong. But I learned about this yesterday, and I'm excited.
That's our like yeah quick learning way of describing what's going on.
Yeah,
I want to stop you earlier because I feel like this is a French name, right? It's like Peclet or
yeah,
something. Yeah.
Peclet.
I don't
I
think we
said
have to say
Peclet, didn't I?
yeah. That scientist is rolling around in their grave.
It's like bonjour, bonjour. I don't know much enough French, but bonjour. I don't know which one's which, but yeah,
Let's motor on.
motor on. So, so yeah, the author set out to use Mips in order to explain this transition of swarms to biofilms. So we've got
Mm,
this like graph here that kind of describes this gray area where Mips is going to happen. I
mm.
think, and up until then, you've yeah.
I think what's important to realize, right? Like you always want to know where this paper, what's the new ground, right? People
Mm,
publish papers because they're trying to tell us something new, presumably. But like as you can see from what we talked about before, there's already a lot known about these things. So like a real challenge about locating yourself in a paper, I think, is like figuring out like what's the new part. And what the authors are saying here is that they've always known that bacteria become biofilms,
mm,
right? And there's been a theory proposed a long time ago, and you'll see this picture over and over again, right? Where planktonic cells eventually like aggregate into small things, and then they get bigger, and then they eventually cover the surface,
mm,
right? And the authors think that that sounds a lot like that could be a lot like Mips,
mm.
right? Like this physical phenomena that's being described with other self-moving particles. And so they're gonna try to use these tools of describing Mips to describe the bacillus moving and forming these biofilms.
Yeah, like this MIPS part, these like traffic jams effectively happen. So when these cells are like kind of moving against them, they can move over each other, and then when they do that, they can form these like kind of islands of just stacked cells. And what I think is being proposed here is that those stacked cells become the basis of biofilms.
Yes, absolutely. And I think importantly, I mean, it almost sounds so minor in this sense, but it's attaching that math that comes from like describing Mips to describing those dynamic Mips islands in this image, right? Like that's like the critical thing I think the paper is really trying to show us. But let's see how they do it.
Yeah. So they've got this half like this kanamycin disc. So kanamycin is an antibiotic. It kills off bacteria. It messes with the 30S ribosome, I think.
Yep.
And they've got like this kind of droplet of B subtilis that they put on the other end of the plate. And so they've let the kanamycin kind of have this area where it's active, and they watch it as it swarms towards the antibiotics. And the idea is that the antibiotics will like kill off the bacteria, and it will overall slow down the swarm.
Hmm. Yeah, this is their model, right, of how it works. And it's already known that antibiotics induces stress onto bacteria and that antibiotics induce biofilm, right? So it's not randomly, right, that they chose this stressor, right? Like it has links already to the biofilm literature. And yeah, they're just going to take a look at it in a way that I guess maybe hasn't been done before. And so yeah, that's the setup that you described in A. And then you can see that the way they do it is they put this disc onto the agar and then they just wait. So agar is like a semi-solid medium for those who haven't worked with it before. I'm familiar with it, well, from the lab, but then in the kitchen from like an almond jello that you can get in Chinese like desserts. So anyways, it's this sort of like jelly-like thing, and things diffuse through agar. So when they place that kanamycin disc down on the agar, the kanamycin starts diffusing through, and they wait 24 hours actually, so that there's a gradient of kanamycin, that the strongest right by the disc, and the weakest furthest from the disc. So as these bacteria are swarming, they're eventually going to encounter greater concentrations of antibiotics. And that's going to be useful because we know that induces biofilm. So they're going to be able to study then something about the biofilm formation in this zone.
Yeah, and so they also show like how that gradient change. So like 0 hours, you can see that it's quite steep as the antibiotic is rushing out. Then it kind of stabilizes at 20 hours, and at 44 hours it might have degraded a little bit, but not by too much. So essentially, throughout the time frame of this experiment, they're arguing that the antibiotics going to be working in a kind of fairly stable way.
Yeah. I don't think it's even I don't think it's even degradation. I think like if you can see 44, that line is like even more flat, I would say.
Yeah.
So like I don't think the dotted line here is not like a limit of detection, I don't think. I think the dotted
that's the MIC. So that's the point
so
where
like
yeah.
Go ahead.
Oh, that's the point where like the bacteria die off. So you'd want to see like all of them being somewhat the same around that area, so that
Yeah. I just want to take a step back because like they cover this in a later figure, but I think it's hilarious how they determine the concentration of kanamycin in this sense, right? Because if you think about it, how do you measure kanamycin in a piece of agar, right? And I think what they're doing is they calibrate the zone of this is maybe too complicated and we don't have a slide about it, but like for those microbiologists out there and for you Faz, right? Because like like the fact that they calibrate like the zone of inhibition from like punches of the agar plate, right, that they extract, and they use that to back calculate the kanamycin concentration. That is just hilarious to me. There's something really funny about like using the bacteria as the measuring stick for the antibiotic.
Right. Yeah. I see what you mean.
I think it's fine. I don't have a problem with that methodology. Right? Like you could do it more advanced. You could like do like an enzymatic assay, right, where you punch it out and you find an enzyme that breaks it down, and you do biochemical proof that that's the concentration. But here in some ways they're microbiologists through and through. That they're going to use microbiology to figure out the concentration of kanamycin at any one point in this plate. Okay, but maybe that's too complicated. Let's go on the paper.
Okay. Oh yeah. So we got the first video where we see the so hopefully this will come out on the stream. But yeah, we're seeing this swarming propagation of the B subtilis, and yeah, it,
yeah. I think we'll definitely play it once more because I do want to point out, right? Like, think of the things that we talked before in the introduction, right? The things we know already about bacillus, right? So you're gonna first see some shady thing diffuse away from the center, right? And that kind of like lightly colored material, that's probably the surfactant at some level,
yeah,
right? Like, I mean, some of it's probably bacteria, but then here around 2 hours, you start to see some density in the center grow. And now we know that our bacteria are growing, they're swarming, right? They're bringing that density out. And they try to get close to that kanamycin disc, but they don't quite make it because, of course, there's like inhibition from the high levels of antibiotics there. And as it goes even further, you can start to see like, oh, it's getting really dense. And then we're seeing some of that wrinkle morphology, right, appear.
yeah. You're definitely seeing a lot more of those like noodly appendages come quite like so. Yeah, so that first front is the surfactant pumping out, I'd imagine, or something like that, and there may be a few small bacteria in there.
Bacteria and surfactant, something like that. But the bulk of the bacteria don't start moving out until you see the density, like the sort of the whitish density.
Yeah, I mean, I feel like it might be because I mean, you see that there's this wave that goes very close to kanamycin disc and then just blanks out. So it might be that there's a few bacteria that get close and then just drop dead, and then the density never gets high enough in those areas. But yeah, it's
actually that's a really good observation. So like, let's play that again because maybe we can see if the front of that actually.
Yeah,
so I mean, this is just paying attention to the first front, right? It just goes.
Yeah, it just goes, and then you see it stopping at this kanamycin disc. So you stop now, and then suddenly you notice that there's a big space behind that first wave. That comes out.
Yeah, so there might be a little bit of bacteria probably mixed in that initial layer, but not too many. And it lays the groundwork for the rest of the bacteria to come.
Yeah.
All right.
All right.
Yeah, that's good. But they're going to do that over and over again, right? Like that is their bread and butter experimental technique in this paper, right? They will do that and they will zoom in to different sections to try to show us the dynamics.
Yeah, and they've got the speed of the front, which is millimeters per hour. So I presume that this is just well, because it seems to be like increasing as it gets close to the disc, and then it stops when about 1 centimeter from the disc. It kind of goes drastically, unless it's a blank. In which case, it stops drastically when it reaches like the edge of the plate.
Yep, totally.
So that's
I think that's good.
Yep. Now we've got another video. What do we have here?
So this the EPS mutant.
Yes.
We saw that earlier too, right? EPS mutant, it can't make the exopolysaccharide, which means that you can't really get as much of the biofilm to form on a regular plate, including on this sort of plate. And you can see here that you still get the same front, right? That kind of gets right to it. You still get the tendrils coming out from the colony itself. Then you get that zone of clearance. You get everything filling in. But what you aren't really seeing is the wrinkliness of everything. And that's because it's missing that exopolysaccharide.
Right. So yeah, the wrinkled phenotype is dependent on the presence of the exopolysaccharide, and they also say on the presence of kanamycin. So when they zoom into that zone of clearance, they actually see these tendrils, these wrinkles coming in there. So I feel like, sorry.
Oh, I would just say that like they say maybe that it's not dependent on it. Like obviously, like when you look at the zoom in with the no kanamycin control, like there's texture there, right?
Yeah.
Like there's probably some sort of biofilm action going on. It's just not to the extent that you see it in the kanamycin group.
Right. Yeah.
That's how I would interpret it, at least. I think, right? Like, like the EPS is so smooth, right? Like, presumably these plates are being incubated, right, for the same amount of time, and so, like, over time, right? Like, those molecules that are being made like are also being, you also have to think living in a group is also stressful in of itself, right? They have this central I guess assumption that they're using, right? Like when they talk about kanamycin is like it's stress that's producing the biofilm in some way. That's a very general term. We know in biology there's many ways to induce stress, right?
Yeah.
And not all ways are equal. But again, like as physicists, right, like we can just say like that there's a more pronounced effect, right, from the kanamycin in producing this biofilm.
Well, I mean, another perspective is that biofilms are more able to survive in that zone of clearance, and it could be that just you're seeing more cell growth in there because the biofilms are more stable. Whereas the EPS mutant, you're seeing less. You're seeing less cell growth because those cells don't have the option of forming biofilms.
Yeah, absolutely. Yeah, you don't even have enough growing there. And I mean, I think one of the things that you said that like you have to remember too, like they're focusing in this specific region, right, where biofilms are forming in the presence of kanamycin. A nice way to double check, right, like if the conclusions that you're drawing from this data is correct, is like you just look at the place away from the disc, right, and do you see the structures, right, that by the disc away from the disc, right, and that kind of gives you like a check on that. So I think they'll talk about it later, but like in that no kanamycin control, when you look at anywhere on that disc, you're going to see that pattern. But in the kanamycin disc, you're only really going to see that pattern when you get close to the kanamycin.
Right. And now they're talking about wavelength and the roughness of the wrinkles. So this is going in deep into their image data. So in terms of wavelength, I think they literally measure the distance between wrinkles. So they take an image and they pick some wrinkles that are near to the edge, and they pick out the distance between them.
Yeah. And I guess they kind of rely on past literature here to say that wrinkles and roughness, like these are things that people have shown in the past to be associated with like a stiffer biofilm, more biofilm, something like that. I didn't dig into some of those references. I know that this is a point of contention because for people in the biofilm field, they may not use these particular metrics to say like when you make biofilm. Actually, I do want to go back to the last slide because there's one thing that we didn't touch on.
Oh yeah.
Yeah, and that is the graph at the lower left hand side or right hand side.
Yes.
In that graph, that's maybe trying to show like another metric of biofilm production, right? Where they're looking at the signal that comes from a reporter from the TAP-A that uses the TAP-A promoter. So TAP-A, as we said earlier, is part of this operon that creates biofilm, right? It anchors some of these, it anchors like protein structures to the surface of the cell that allow the exopolysaccharide to accumulate. And so you would expect that you would get that turned on if you were making biofilm, right? And if it gets turned on, it would make a YFP signal in this case.
Yeah, and in this case, they see that in the no kanamycin group, I presume that they don't see any TAP-A, whereas in the kanamycin group, they see quite a lot of signal from that. So that implies that they are making lots of biofilm in that specific plate, which is
we don't see as much in the TAP-A right condition. So like if we were really strict geneticists here and we were trying to get genetic information from this particular graph, right, we would say, oh, they're missing a control, right? They're missing they're missing a way of disrupting the promoter itself to show us the flat line, right? Because like even in no kanamycin, you can still get reporter signal to be instigated, right? And so like at different levels, that reporter could be creating biofilm, right? And that's why I think that it's very important to think of, like the biggest thing to be wary about in this paper is what does it mean when they say make biofilm? Right? Make more biofilm. Like biofilms are like it's just any group of bacteria that come together and make a little bit of this matrix, right? Like really they're describing a difference between the quality of the biofilm, I think, that's being created, and they've focused very closely on the quality of biofilm that's induced by kanamycin. So just to put that out there, right, as a good way of limiting our interpretation. This is not all biofilms we're talking about. We're talking about a specific form of biofilm that the authors have like anchored on.
Yeah, I think that's an important point. And so they also
more roughness, a bit greater wavelengths.
Roughness basically like they take a transect and they look at the kind of difference in intensity between pixels along that transect to basically say, okay, that basically relates to the height of the of the biofilm or the texture of it, and that gives the roughness. So therefore, the idea would be that the rougher the biofilm, the more like knotted and weird the biofilm structure is.
Totally, totally.
And figure S2, so uh, more
The more kanamycin in the disc,
yes,
more wrinkled the colony.
yeah.
So again, I think this is really focusing on down. This is helping us focus on the phenomena that they're studying, right? They're studying this very wrinkled biofilm formation, right, in the presence of kanamycin.
Right, yeah. Uh, and without kanamycin, even after a week, they waited a long, long time. They didn't find anything that's closely resembling wrinkles on their no kanamycin plates.
So this is very much in response, right, to that sort of potential criticism that could come up by saying like, oh, but you're going to get low-level biofilm everywhere, right? And yeah, you do, but not like the wrinkly type.
Yeah. So again,
right?
Yeah, because some people because I would define this as a biofilm, but it's clearly not the same structure or like phenotype as the wrinkly biofilm. So
exactly.
So it's a kind of looking at differences in kind rather than So
rather than absolute biofilm, and I think that gets washed away sometimes when people read quickly, right? Because like that's the devil in the details of science.
Yeah. So I mean, I feel like if I was a biofilm specialist, there's a lot of things I could nitpick about this paper, but just that'll be just purely in the language of how we're talking about it rather than the data itself.
Yeah.
Okay. Click, and now they use hard agar, which is agar that's too hard for the bacteria to swim through.
Yeah.
And so they've dotted some colonies onto there, a certain distance from a kanamycin bump, and they found that colonies that are closer to it didn't have like they had a very flat morphology, and even the ones that were kind of far away, you can see that halfway through that they suddenly stop having wrinkles, which is actually quite interesting.
This is so cool, I think, right? Because I think that to me, this sort of shows the particular space that they're trying to figure out what's going on, right? Because in this circumstance where you don't have bacteria swarming around, the kanamycin seems to inhibit the wrinkles. But like in the other circumstance, we've been saying that more kanamycin, more wrinkles. So there's obviously something going on here, right? And that's how they describe it in the paper as well. But like there's something going on here that requires the bacteria to be in this like loose state in order to see the types of phenomena that they're going to be describing in the paper.
Yeah, I mean, it could be in this paper that because I have to like say the opposite. The non-believer would say that okay, but here you're basically getting less bacteria growing inside the area, the zone of inhibition, which therefore means that you might not have advanced biofilm development. Which I appreciate means that kind of I'm having it both ways when because in the opposite glass paper I was describing the complete opposite of like oh yeah, biofilms are more likely to survive in this. So it's. So, but I think there is something definitely happening here, and it's quite interesting.
Yeah. I mean, okay, there's a way, like, they're not asking that question, but there's probably a way to try to disentangle growth from this phenomena, right? They haven't done it in this paper, but there might be a way to do that, and that's not exactly what because they're not actually going to tell us about growth necessarily. Growth is always assumed to be happening in this whole paper, I guess. Yeah, growth is assumed to be happening at a certain point. Anyways, that's an interesting point that you bring up.
So yeah, there's and I think this is quite a very interesting one because this is where they look at very early on. Did I get this is the right video? Okay, right.
Let's go.
Yeah,
it's going.
Yeah, so looking under a microscope at swarming bacteria and looking at like different time zones and looking at different densities. And
wait. Oh, that was fast. Let's show us the previous one again. So the idea is that the one on the left is from an earlier time point and one on the right is from a later time point.
Yeah.
That dark spots are density because like as bacteria clump up, you can they I guess inhibit more light traveling through them and you get little dark spots.
Yeah.
And so if you really focus on the left-hand side, we're gonna have to play that again, I
Yeah,
think.
we're gonna have to. Yeah, definitely. So.
The authors want to say that there's these flashes of dark spots that are appearing but disappear, right? A transient density that's forming, right? Transient traffic jams is what they're trying to allude to here.
Oh yeah, you can definitely see those like popping in and out in the earlier time point, and then they become
Yep.
more permanent traffic jams at a later time point. Uh,
Right.
uh, where yeah, those seem to be like just clusters of bacteria, and I mean it's hard to see at this resolution, but I think what the researchers are presuming to say are that there are bacteria coming in and out of those traffic jams all the time, but the
Yep.
clusters themselves will remain.
Yep. Eventually, at some state, the clusters start to gain permanence. And and remember, this zoom in magnification. This is on one of those regions of interest, right? That that's close enough to make the wrinkly biofilm. So what we're seeing, right, is from a very early time point, right? What's the journey for that region to become the wrinkly biofilm?
Right.
Okay, I think
Okay.
that's
Yep. Let's. So yeah, again, we're now zooming in even further, so you can literally see the cells that are going in and out of this traffic jam. So the darker area is this island where think. Oh, sorry about this. So yeah, where we've got like the dark areas are like multi layers of cells. So they because they're stacking on top of each other, they inhibit more light. And yeah, I feel like I've been
you.
here in the mosh pit.
Can you turn on a loop?
I let's see if I can. Uh,
Doesn't look like it.
it doesn't look like it. I'm
No, it's fine. Don't worry about it.
yeah. Hold on
Yeah, but in this one, I guess they're trying to see that the cells in the dark areas, they're still moving.
yeah.
I think is one of their big points, right? And actually, they even think that you can see. I can't see it. It'd be nice to see some particle tracking, but like you can see cells from the outside move to the inside, right? From the light area, move into the dark area. From the dark area, move into the light
I mean,
area.
I feel like if you look at the top part of that, you can sort of see a cluster of cells just move out. So like, oh, you suddenly see like
Oh
clusters move
yeah, I
out,
do. Yeah, I think I see that.
and you can sort of see some cells moving in and out. Like
Yep.
so some of them are just bouncing off the surface. Some of them are going in and out. It's yeah,
So again, an important observation, right? Because these islands, even when they seem permanent and they're sticking around, they're actually dynamic, right? Things are flowing in and out of them.
yeah. Uh,
So it's a local slowdown. I think the traffic jam mentality is such a good analogy to bring through this whole paper for
Yeah,
people.
because I feel like a mosh pit analogy would be a lot more harder to talk about. But yeah, I do feel like there is a circle pit kind of thing going on with those bacteria. But
Actually, mosh pits are not bad because like you do get like I've been in mosh pits where you kind of get like moved around in a certain way. You don't have that much autonomy, even though before you did, and then you can get like pushed out of that circle or go back in.
yeah, and there's like the wall of death where people are organized to go on one, and then there's the areas where like suddenly everyone backs away and there's a bunch of shirtless guys hitting each other, and you're like, okay, well we have to find a way to move around that somehow.
Yeah, that doesn't this physics it doesn't describe the areas of clearance.
And I'm so disappointed by that. Physics, come solve the problem of mosh pits. But yeah, this looks at
So
this. Yeah, sorry.
okay, go ahead.
Oh no, this looks at the speed of the bacteria as they're going to. So almost looking at this is where the particle tracking goes in. And another thing that's quite interesting about this paper, they don't really talk too much on the methods, and I wish they did talk a bit more. Is about the image tracking software, and because literally for about the last 10 or 20 years, people have had the ability to put a video camera on cells, but only now have we had the advances in particle tracking, advances in video, like image understanding that allows us to do analyses like this.
Totally. Yeah, to say like here's the same bacteria and then you basically I think they don't show you any image of this, but like the idea is that you track it so it makes a little line
Yeah.
right over the over time. And then the computer can take all of those lines and average them together, get things like speed and yeah, general direction sometimes as well.
Yeah, they look at the cell speed and they basically find that inside these islands, they call them, the speed of cells is much slower, and outside the monolayer, it's much faster, which kind of really, really tracks with what we saw in that image. Where I'll see if I can bring it up again. So in this, where you see like inside these cells are very like stuck, but outside they're zooming around. So it is very much like a traffic jam.
And again, just to reiterate for people to understand, like this data is coming from many times they've done this, right? And taking pictures, different regions of interest, and then they average it all together, and you get graphs that look like this, which is great, actually. It's so tight, the data here, right? I find the data very tight in this sense. 0 minutes is the time with those permanent islands forming.
Right.
So you can see at negative 60 minutes to that phenomena, the cells are just going crazy, right? They're like zooming, and we saw that in the earlier videos as well.
Yeah, and as they go closer to the disk, you see that zooming slows down a lot. So they don't measure like the distance to the disk like in the early phases. I'm presuming because the cells haven't reached those phases yet at that time point. But as they get to the 0 minutes, they suddenly see like, okay, cells are slowing down a lot.
Totally.
Oh, and we've got oh, this is interesting. So we've got a broad view of like how the so this is actually like a very processed image. So they took a camera photo and then they did some let me see. They just did like an image algorithm that I know I've wrote down somewhere that but local adaptive thresholding, where they take this is a way to kind of enhance the contrast of an image. So essentially, like you've got, so you convert. So
play it first.
Let me see if I can describe this, because again, this is very much a physics-y, like kind of image, computer vision-y kind of understanding of things. But I think the best way to say it is like if there's any like difference or any kind of cell boundary, it will pick it up. So the idea was they're using this to pick up the images of the islands when they first form. So
yeah, I think like it's nice to like when you said thresholding. I'm not sure if people are familiar with like it's like you're trying to make something that's a continuous into like into more discrete steps, right? So like the idea is that, oh, like if you get above this threshold, right, of grayness, actually you're in this category of color now, right? And the way that manifests in this particular video is that anytime you see like density grow, like when you see close to that disk, you see like there's actually these shapes forming.
Yeah.
Right, and that's like initial density, right? And then you'll see like eventually all of that merges together and you get an even darker version appear on top of that, right? So like that means that it's getting even denser, and it's actually interestingly in the same pattern. And then it goes again and it gets darker, and so like that's what the thresholding is giving us. If we were to see this without thresholding, I'm not sure if we would see the patterns as easily. It would definitely be busier because you would see all that motion. Maybe that would be part of it.
Yeah, you wouldn't see.
We're a lot more zoomed out actually, so maybe you wouldn't see the motion.
I mean, the interesting thing is like, imagine each of these gray spots is like an additional layer. So every time it gets dark, you've got an additional layer of cells. So what we're seeing is like near the disk, suddenly in the first initial, we see these dark striations appear, which indicate there are some multiple layers of cells occurring there, and that eventually merge together into like a full layer. And you can see that it's got. So like there's this area of like extreme darkness where there are these really thick layers building up.
Yeah. And just to remind people, like it's happening even far away, right? On the other side of the disk, right? Like this is the place where there's no antibiotic, right?
Yeah.
And this is just happening because of growth, right? Like we're just getting growth. And you can see it forms it's spotty, right? That second layer comes in spotty, and then it gets darker, right? And like it's kind of interesting to think about this, right? Like there's nothing stopping bacteria from becoming dense, right, in different areas. Something about these antibiotics, right, is promoting a certain type of behavior.
Yeah, this is really fancy because you see like this area of like, so this bit will get thick very fast. You've got these striations, and then this bit in the middle will still stay quite blank. You see some little dots happening. There's more bacterial growth naturally happening, and then you see this merging of layers, and then this bit becomes just a lot more darker.
starts growing even in the same pattern, which I find fascinating that that's happening, right? Like that in the next layer. Well, the thing is, it's not precise, right? Like
Mm.
next layer, right? What does that really mean? Like it's not like we know that their setup gives us resolution on like the number of layers of cells, but like still, there's something very intriguing about seeing the same pattern appear twice.
Yeah. I mean, that's one thing that I'd like to have seen is if they did do like some, they could actually like correlate those pixels to actual cell numbers. So just by taking the layers at specific points and then doing some cell counting, and
Hmm.
then having that a nice little like line that we can use to kind of feed off and extrapolate cell numbers from. Um,
I don't know how they do it because it's so small, right? Like these are small features, and they're happening like at a specific time point.
yeah,
I think there's probably a way to get there. I just wouldn't. I don't. It would require some cleverness, I think, and I think it would be done with reporters actually. At the end of the day, I think that's a
yeah.
really part of it. Um, like, yeah. Sorry, I'm just now my mind's going crazy when you ask that. We'll talk about it at the end.
Yeah,
Let's
that's the thing. This inspires a lot of imagination, which I quite
mm,
like. Um,
mhm.
yeah. Oh, there.
So this is just like the static images of sort of what we saw, right? And showing that that initial thick area, it actually occurs at a set distance from the disk, right? Not too close because you guess bacteria can't grow that close. Not too far away because apparently kanamycin concentration is required for this particular phenomena, right? It happens at this like sweet spot, 0.7 centimeters away.
Yeah, and we literally saw that in action. Now they just put a graph on it, and this kind of gives confidence because we only saw like one cell, like one plate, so we don't know whether like that would happen with other plates. Whereas the
Totally.
hope here is that they've replicated a couple of times and they found that there is a standardization happening.
Yeah, yeah. I'm
Uh,
just like flipping to the figure potentially. Where do they say how many replicates it is? Do they say how many it is?
I don't know how many replicates it might, because this is a difficult thing in this paper because it's hard to
No.
determine like whether there are
This one is not that what we showed and figure three supplement three, not replicates.
not replicates.
That that's just quantification of what you're seeing.
Right, I see. So, if anything, they're pseudo replicates, or so that kind of weakens it a bit. But I mean, yeah. So, we can talk about that a bit further. We're now on Figure 3 ABC.
Figure three ABC is a summary of that whole image, right? Like, but they're gonna now take their I guess their more strict understanding of what's going on and try to apply it onto the time scale, right? So
Mm,
at the beginning, right, at like before you get those dynamic islands forming, that's an hour before. It's a monolayer, presumably. Cells are not so dense; they're moving around. Then you start getting these like local traffic jams that form, but things are still moving in those local traffic jams. And after that point, you start getting those, you get layers on top of those layers,
mm.
right? And those layers start merging together, and you get layers on top of that. And they use this rho C to talk about a density of cells, right?
Yeah,
So they fully understand that the cells are growing over this time course, right? And the growth of cells increases their density that is leading to these different behaviors.
yeah, and so yeah, that's sums it up. I think we can move on to let's move on to the next part, which is Figure DE, which this is a way of them to try to characterize the motion of these like cell islands. Um,
Hmm.
So yeah, when I first saw this, I thought, "What stain are they using?" But not using a stain. This is just looking at the image. So the bits that are in like dark are the bits that don't move very much, as far as I can tell. And or is it
That
the?
appear first, I guess.
I think that
Darker appearing first. Oh, I'm
yes,
not
I
sure.
think that's right. And brighter appears less later. Um, so
See what this does.
but I think this is a good way to show those weird striations, the way that the islands are patterned. They're very
Hmm.
elongated and almost towards the kanamycin. So I feel like in this sort of paper, you would expect them to be kind of like kind of right angled to the kanamycin. Like you kind of imagine these cells stopping as a barrier rather than creating these lines. Um,
Hmm.
So it'd be
The idea is that the features of the image are being colored by the time in which they appear. So
yes.
darker ones appear first. So actually that is what you were saying, like things would appear further away. There you can see in the striations, it's darker to one side than it is to the other. I would say it's darker in the middle, where between the transition from dots into lines.
Oh, I see.
That's my reading of this particular figure. And so like those places, those features form first is what they're saying. And then it starts like getting closer to the kanamycin.
So yeah, it's almost like these islands form randomly like dots, and then they striate out as they grow. So almost like as
Yeah.
as you go into
I
the
I
yeah.
do think part of what they might be showing here is the growth of those islands. I think that that's a really good way to think about it as well. Like
Mm,
whatever these structures are, they start in a certain sweet spot and they grow out that way. There's a lot to describe here, right? Like
yeah.
uh, you can even I think from the physics perspective, like in some ways, well, like you could ask that from a purely physical perspective, right? Like quantify that for us, right? Quantify that
Mm,
across many different islands, and then start varying things to understand that relationship. Again, this is more the first report of attaching all this Mips language to the phenomena, and yeah, maybe not first report. They've done it before, I think.
yeah. So uh yeah, these oh, this is a test where the quorum sensing has an effect.
Yeah.
So yeah, because quorum sensing is a very important bit in how bacteria communicate with each other and coordinate behavior. So if this is coordinated behavior, does changing the quorum sensing have an effect on it?
Yep.
So
They show that it doesn't. They still get the same wrinkles. I would say that those wrinkles look different. They didn't do roughness. They didn't do wavelength. But yeah, it's still
yeah,
making the same structures, right? It's not the same as making the EPS mutant where they can't make the structure at all, right? Like removing quorum sensing is not such a strong phenotype here.
yeah, I feel like that I do feel like I'm seeing some differences, but I mean, from a physics perspective, you really don't want quorum sensing to be a major factor because that harms the predictability of this model because it's one of those things that you can't necessarily adjust for. Um,
Yeah, I think that when I see this data, I say, okay, it doesn't matter in terms of forming the 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 wrinkly phenotype, but it's doing something to the wrinkles.
yeah, the
And so, like there's something else at play here that isn't included in the model. But okay, let's.
but yeah, you'd kind of expect that with bacteria because they are living things and something odd will happen. So I mean, I feel it's good that they've got this background that they can eventually come back and look at later. But this, I think, is the real like kind of big. Sorry about this. Big money part of this paper.
Mhm. Yeah, so here is where they're gonna go back and they're gonna say we have all this Mips language, right, where we can describe the formation of mobility motility induced phase separation, and specifically they use rods in a 2D space, right? So that's their assumptions that they put into their algorithm, and they say for across all these different Poclet numbers. Which is proportional to the speed, roughly. And for all these different densities of bacteria, surface coverage, right? Where would we assume that that these MIPS events would happen based on the prediction? And so that's the gray region that
Mm.
they shade. And then on top of that, they've seen many different sections of an agar plate where they can measure how dense it is versus how quickly the cells are moving, which then informs this Poclet number, and they can plot that over their predictions. And they can also then say where in those predictions do they see the jams actually occurring, and you can see, oh, they're getting pretty close. There's something here, maybe.
Yeah, I feel like this is, and this is going back to that phase diagrams we saw before, like where the this transitioning from this liquidy form to a solidy
Mhm.
form. The only thing that I feel is missing is actually like some statistical model comparison, which is what I'd kind of expect when you have this a model this kind of interesting.
Yes, I see what you're saying.
Yeah,
Yeah. So what's the agreement between the predicted, right, from the jams inside of this region for the jams outside of this region, right?
yeah, and also like seeing like whether there are any specific places where that it's like digressing from that, because it's kind of interesting when you're looking at a model and you see something where a point where everything agrees with your model is great, but even more interesting is where it digresses from that model, because that can be an
Sure.
interesting avenue for research later. And
Yeah, I mean, if at every point that they've collected, they also have associated metadata, right? So like they have things like the condition at the time, the temperature, all these different things, right? If they had metadata associated with every point, then I think what you're proposing is incredibly powerful, right? Because it would mean that they could start to try to pick out, right, like the reasons why these things differ. But yeah,
yeah,
right now it's more of just like a proof of concept, and you could definitely know
yeah,
that if you ran some sort of like how many red dots are inside the region, right? How many red dots are close to the region versus how many blue dots are far from the region, you would get like a some rough quantification of the same thing.
yeah, because again, that region is almost like generated almost in silico, where they don't really have all the information. Whereas,
Oh yeah, I don't think even almost that region in gray is purely theoretical.
yeah, so actually getting input from this like real life situation could actually feed back into the model and tell you something about the situation in that petri dish, and that could help you think about other 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 underlying factors in this. So
Totally,
yeah,
totally. Already thinking like a computer scientist.
I mean, I always try to fit. If I can fit a line to my data, I do so because it makes things look more fancier, and
Huh?
and then I can pretend like it's a law rather than just a random collection of data points. So be like, oh, it's a straight line, and therefore it's directly proportional to something. Could be, but it's biology, so it's directly proportional to something until that thing reads your research and goes, no, no, I'm not gonna do that anymore. So, I mean,
Until you find the covariate, right, that pulls out some of the noise.
yeah, um, yeah, and this on in B you've got this speed, and it kind of has this almost two phases where you've got the speed normally, and then when it's in these jams, it goes right down.
And we've really seen that before, right?
Hmm.
Like when jams occur in a region of interest, the speed is down.
Yeah,
I don't know why this was in this particular figure. It's the same as we saw, I think, in one of the supplementaries.
yeah. Um. Oh, and this. So they used antibiotics, but now in this phase they're using UV radiation. So to
So they still have, I think they still have antibiotics in this particular experiment,
oh
right? It's just that they are also applying UV radiation.
oh really? I assumed it was just UV radiation, but
Oh, is it just radiation on a plate?
yeah.
Um,
So in this
yeah,
plate, yeah.
that actually think I think you're right. Yeah, it is just yeah, this is because antibiotics was one way in order to stress out the bacteria. Now they're using a different stress.
Yeah, you want to have different ways of inducing stress. If you want to say, if you want to indicate that it's stress that's doing it, not just this antibiotic that's doing it, then you can find another way that causes stress, and then see how that response changes. So
So
in
the big jump maybe here is that they initially set up all these observations were predicated on this little toy example of the kanamycin inducing this phenomena.
hmm,
But now they want to focus in on the graph actually, right? They want to see like how can we show that this graph is actually representative of what's going on. And so maybe not kanamycin now, but if they could do other things in the graph, right, where they're just manipulating surface coverage and Piclet number, and they show the phenomena occurs, then that's good evidence that they're going down the right path.
right. So in this case, they irradiate an entire plate of bacteria to show that they all stop, and
All
and
slow down.
yeah,
Yeah.
they all
Oh, they
slow
do
down.
actually stop. In 3 minutes, they do stop.
Yeah, so some of them stop, but they mostly slow down. And you can so in the first image, they're showing like, okay, we've got this, and inside that they've got their field of view. That's the bit they're looking at. So that'll come in useful later. So put that in the back of your mind. Second part of that is the surface coverage over time, and uh, finding that actually that decreases. Well, no, surface coverage goes up, but the speed decreases. The speed is in blue, and the
Yeah.
point where they put on the UV light is in purple. So
Hmm.
you can see that it kind of goes up a bit, and then it just like goes right down.
Yeah. And the way that translates, because like again, they can measure right the surface coverage in the region that they're looking at, as well as the speed of the particles, which gives them the Peclet number, and they can show you then in B right that the dots that they start with after they irradiate it, they all kind of lower in speed, so the Peclet number goes down. There's maybe a slight surface coverage increase, right? Very slight
Yeah.
to the band, but that particular intervention, right, isn't predicted to promote Mips, and it doesn't.
Yeah.
So that's good. So far, the model is holding up.
And where they take this quite interestingly is where they try to create their own traffic jam. So they pick just one section of the plate, irradiate that, and then watch as all the other bacteria bump into those irradiated bacteria. So
Yes.
So yeah, they kind of do their UV light and they like kind of zap some bacteria, and some just do genuinely stop, but the rest of them just bump into them, and then they form these multiple layers.
Mm-hmm.
Um,
So is this image actually with UV though?
oh well, let me double check. I could be wrong about
I don't
this.
think it is. I think that this is just these are being irradiated by UV light.
Yeah. So.
And the UV light is as large as the field of view. Okay.
Right.
Yes. So yeah, so now instead of irradiating far away, right? They're just irradiating the place that they're looking at. Right?
Yeah, and you can see that there are bacteria flowing in and out of it.
Mm-hmm. Mm-hmm.
And there are some
down, and yeah, it's getting denser.
Yeah.
Cool.
Cool.
So what it was here, right, where now they've just irradiated the section that they're looking at, and as you described, everyone's bumping into that slowdown zone, and in doing so, they eventually jam up, and you can see that this is how it looks, right, with the speed, and yeah.
Yeah, and interestingly, they also show like kind of the transition of individuals into this area of Mips.
So actually, in this way that they're manipulating not so much, so like the speed actually in the view that they're looking at, it goes down just a little, right? But actually what's happening more, what they're seeing more, is all the other cells from other areas joining into this cluster.
Yeah.
Yeah.
And only when enough cells have reached a threshold for it to become really crowded does the speed go down again.
Yeah, exactly. Yeah, so like through localized speed decrease, right? They're able to increase the surface coverage in that area, because that's what they modeled, right? That's how they mapped it out on their map in D, right? Like you don't actually see a lot of speed going down, right? Probably because they're not irradiating a very large area, just a small area, and they're taking the average speed of everything around it.
Yeah, so effectively they're creating their own little car crash and 20 car pileup for the bacteria.
Yep.
And they're finding that pileup forms almost like the solidified cluster that can be described as mobility-induced phase separation.
Absolutely.
Yeah. And now they use a physical barrier. So on one side there's like a hard like wall that these bacteria are running into. And that causes them to kind of stack up and form these multi-layer islands. So you can see these like shock waves of layers forming from the edges.
Yeah, I find this is very interesting to me because it's like they're really trying to show that it's like the density it's that increase in density, right, that is causing these things. But very specifically density during swarming, because in the solid plate, right, it didn't really. Yeah. I'm kind of, I think through the whole thing, I was just like, they'd never used any swarming genetic mutants. I think that's why I put it in the introduction, because I kind of like was hinting towards they did other genetic mutants, but they didn't do the swarming genetic mutants.
Right.
But they wouldn't move across the plate. So like, how would you analyze it in that? It's just like they would need a different experimental setup to figure that out.
Yeah, they'd have to like artificially induce the flow on the behalf of even though if the cells can't move, you have to find some other way to move them in order to model the same island formation. So the idea would be can can you artificially induce these islands through fluid dynamics and then see
Yep.
whether that, and that is a very difficult experiment to do. So I can understand
It
why
is a
people,
very difficult
yeah.
experiment to do. That's why yeah. But that would be really interesting. I mean, I would never ask that as a reviewer on their paper for it, but like just reading the paper and thinking about what comes afterwards, I think that would be really interesting to test some of the ideas that they're discussing here.
And I think that'd be quite interesting in terms of actually fluid. I mean, I guess we'll talk about this afterwards, but actually, like how we handle biofilms in like fluids and how they develop. Uh,
Okay, let's motor on.
yeah. So this again shows that. I mean, you can see here that there is a stark wrinkling when it comes up to the barriers. Um,
Yeah.
and there is a slight like kind of. We've kind of seen it a little bit in the previous things, but there's slightly around the sides of the petri dish as well.
Yeah. I really like this particular intervention because because it's physical, right?
Mm.
It's lending a lot of credence for me to the particular scenario. Like it's like this aggregate, not to say that the aggregation can't change gene expression, and I think we'll see that a little bit later, but like it's purely that feedback between the physical scenario and this particular phenotype.
Yeah, I mean, I feel like they could have done this in with all the previous images as well as almost like comparable because you do
Yeah.
get something like that because the petri dish has barriers within it. So I feel
Hmm.
like they they could compare this and try and pass out what they do. I think they do like because the great thing about this is they posted the reviews with this paper as well. So they do get into it in the review section and they do kind
Yeah.
of justify why they didn't or why the edges at the side look slightly different to the edges. Basically, because the way agar is created, the edges around the agar are slightly different. So it's almost like
Yeah.
there's like a meniscus that forms that is like a lip. So it almost like so it's less like a wall and more like a steep hill.
Yes. Yes. But here they actually like carved a block out and they placed it on the surface of the flat agar. So
Mm.
you get this like yeah, the really like sharp cutoff in that zone. Okay.
Yeah. Okay.
Oh, and here's that thing that I think is bizarre,
Mm.
actually. And really, they're not doing a lot of follow-ups with this type of understanding, but they show that that Tap A reporter actually has this jump in expression all across the plate when they add in the barrier.
Yeah, I feel that this almost feeds in. I mean, I feel like this feeds into the idea of shockwaves of biofilm expression. I'll go back because when these this wall of so the idea that you're getting not just like multi-layer formation, but you almost get like this like the traffic shockwave where you get more like of these like islands propagating further back because of this barrier and
Yeah. Well, yeah, to me it means that the message is being passed through the bacteria. I
yeah.
immediately think like, so what about this one with the quorum sensing mutants?
Yeah, that would be interesting. I feel
Right? Or like or something like there's some sort of communication, right? Because it doesn't make sense that like across the whole plate, even where we don't see the phenomena occur, that the gene expression of this the reporter would be this way. Um,
I mean, I feel like they almost had this a similar thing with the kanamycin. I mean, I don't think they looked at the plate itself, but I do recall like looking at those images. There were more striations on the kanamycin plate, so almost, so I mean, this is a very weird sort of thing to see, and I don't think we have a great explanation for it, but
but in the kanamycin plate, you can say that the ones further away may still have extremely low levels of kanamycin present, right? Like they're
right,
still ultimately seeing that molecule. But here, there's no molecules, there's no additional molecules besides what the bacteria are bringing to the party. And I think that's what really intrigues me about this this particular piece of data that they don't really follow up on,
yeah,
anyways.
because I think that there has been talk because I didn't really dig deep in this, but there has been talk about like almost of B substance behaving multicellularly at some points
Yeah,
through signaling and it could be something like
absolutely.
that like
Mm-hmm.
but yeah, as you mentioned through quorum sensing, I mean, I think the really outside of the box crazy explanation that I want to be true just from this paper is the idea of like this physical process being its own signal. That'd be a really interesting idea.
Yeah, no, I mean, I think it is. I think that that's what they're trying, that's what they've essentially shown with this, right? Is that the physical process is its own signal.
I mean, I don't
But
think
signaling, What?
I
I
think
don't know.
they've explored that as a possibility, but I think without doing the quorum sensing, without doing other sorts of control experiments, we can't say that for sure. So,
Oh, I see. I see. I guess I'm saying that it's a signal. It could be acting through quorum sensing.
oh right, yeah,
Yeah, yeah, yeah. I wasn't thinking like, oh, I see what you're saying. Like more direct, like physical, directly, like has like mechanoreceptive properties, right, on the bacteria, something like
yeah.
that.
But I mean, I think the idea of because I mean, this is similar to like lipid rafts, like where you get these like kind of bacteria clustering together, and because they cluster together, they can signal together as one, and therefore quorum sensing molecules will behave slightly differently because that will be treated as like one almost meta-organism, and then yeah, it'll
Yeah,
be
because I think that that's something that people forget, and we have not done a good job in describing the background of this conversation now. But like people forget that in quorum sensing, that it's not just like a broth culture that's coming up to a certain OD. That in real life, in bacterial living in the open, those clusters are far smaller and localized,
mm,
right? And so like those interactions aren't just like everything's coming up and the signal's turned on, but rather the signal gets turned on in certain regions that then like have interplay, right, with other signaling pathways.
yeah, for sure. I mean, the thing that would be interesting is also like the time to tap a signal as an idea of itself because I feel like if it's closer to that barrier, the time the time for the peak signal should be quicker than in the other areas if it is something like that. So I feel that'd be an
Mm.
interesting thing to look at. It might out to be nothing, but
All right, let's see what's next.
yeah, right. Oh,
Oh, this is they're gonna bring it home now. Well,
yeah.
well, what they perceive as bringing it home, but like I couldn't read. like
I
I actually couldn't parse any part of this as like something useful. Um,
Yeah, I yeah. I mean, if I were to read this like without seeing the reviews, I'd have said, "Oh, this is some idiot reviewer forced them to do this experiment or something." But
yeah.
now that I see the reviews, I'm like, no, they decided to do this on their own. I don't quite know why.
Yeah. I think, I mean, to me, I think it's because they really do have some latent hope that their technology will have, their discovery has some sort of application in therapeutics.
I,
That's my thought.
I mean, I feel like
Uh,
this is like the last figure syndrome for a lot of papers, where you get this lovely story and then they just go, okay, we need to make it look like it's practical, so that it sells, gets citations and stuff. So people,
yeah.
those antibiotic people, they're always citing stuff. So let's put this last thing in. It'll be really fancy. It'll be a really good way to end the paper. And so
Yeah.
I think we've come across a lot of papers now where we've seen like this wonderful story and the last figure just makes no sense because they're like, oh yeah, and it's related to this random thing. And like, okay, you do you. I was with you up until this point, and I'm still with you. That's gonna put this to the side.
Yeah, so what they do is that they think, oh, well, they know that there's like a region, right? Like there is a region that didn't really make these crazy wrinkled biofilms, right? Like
Mm,
you saw that the wrinkled biofilms only occur at that zero point 0.7 centimeter distance, and there's this other region that doesn't get it. So maybe it's because of the concentration of antibiotics there is the sweet spot, right? To like to be able to inhibit biofilms. So if they were able to somehow create like waves of antibiotic concentration going over their bacteria, right? Those waves, when they get desynced, the sweet spot of inhibitory concentration will pass over the whole disk and we'll get less biofilm. That's I think that's how they're thinking about
I
it.
mean, I feel like they're trying to disrupt the Mips formation directly, almost. So they're trying to target at the point where these like islands form. They're gonna try and add an extra level of antibiotic at that particular time to disrupt those rafts and therefore to disrupt later biofilm formation.
Yeah. Yeah, but like that MIPS formation is like at a very specific area with a specific set of antibiotics, right? And so like that's like a stable scenario where the bacteria are allowed to adapt to their scenario. So they're like to me the logic behind going down this path is saying like because that area is stable, right? If we make it unstable by just like applying antibiotics again, right? Then maybe it won't form.
Yeah, so I'm gonna so I went in there and they do list like the distances where the antibiotics are, where the actual like zone of inhibition is. So this is like the
Ah,
region where the bacteria, the edge of the bacteria and the blank space around the antibiotic are. So
Hmm.
so I penciled these in on the graphs because I was kind of curious about. So they let them grow up to a certain level, and then they add a second round of antibiotics, and you kind of see that the actual differences in concentration can be quite big when.
Yeah,
So
totally.
in this graph, I feel like they've proven that antibiotics can stop bacteria from growing, which is
Hmm.
good. But because I feel like, so let's say you've got like, because they're always looking at the edges of the interface between the area where the biofilms are forming, are the interfaces between like the minute where the bacteria will die off and when they're fine basically. And then
Hmm.
by adding another layer of antibiotics, they're disrupting that, and they're changing that to suddenly an area with lots of antibiotics. Um,
So describe to me what these lines are again that you've
so
overlaid.
these are where the back. So let's say after the first. So just before the second dose is given, this is where the
Hmm.
edges of the colonies are. So this is where the
Ah,
biofilms
got
will be
you.
forming.
Understood.
So
Yeah, but the data that yeah, so like I think it's important maybe to describe like this goes back to that very beginning thing where it's very esoteric the way that they've figured out the concentration of the antibiotic across the whole plate, but they have two plates, right? One plate where they put the bacteria on, the other plate that they're just harvesting segments of so they can figure out the concentration gradient that formed.
mm,
um, and so, yeah, I mean, it makes sense, right? Like in the ones where they put a lot of antibiotic at first, right? The the bacteria stay away further, right? And so the region that they're going to look at has to be calibrated. That they're going to look at the further region.
yeah, so you've got this region that has like that forms an area. So it forms like a halo around. So you've got your kanamycin on one end, and you've got like your bacteria growing, and then these
Yep.
bacteria they're fine with this concentration. Then you kind of up the concentration by adding your second dose, and suddenly
Yep.
you've gone up quite significantly. So those
Yeah.
bacteria will probably be arrested that phase of growth.
Yeah.
Um,
Me, I'm like, yeah, it's like it's interesting. It's weird to me. I guess I don't really see it as if the theory here is that they want to see a disruption of Mips, then I think that they have to collect data that is as dynamic as Mips. They have to look in that region and they have to count the number of those transient islands that form or something like that,
mm,
right? Because I think that is the better, like I think you're giving them uh yeah, in discussing this and discussing it with you, that's the better measure.
right.
Because it's not really whether or not the biofilm is gonna like their hope, I guess, is to see the biofilm just totally abrogated by this strategy. But as we'll see in their data, they don't actually see that. So let's move
Right.
on.
Yeah. So moving on to the actual data where they look at so this was like your 200 very much like
Yeah.
their controls, and then as they go further they find that the wrinkles they don't exactly go away, but they seem to their pattern changes.
Yeah, yeah, and remember that these images are being taken at different locations
Mm.
across the plates, right? Because they have to go to the region, right, that they know are going to form the wrinkles.
Yeah, so just going back, so like uh
Mm,
the
mm,
in yeah, you can go see in the corners that they get closer and closer, and then
yeah.
they get washed over by antibiotic. um
Yeah. So, yay, I guess is what they is what they conclude from seeing this data. But to me, it's more interesting to just know like they've set up this really interesting model that there might be, as you said, Fuzz, there could be this right? It's very interesting to postulate about some mechanoreceptive, right, thing going on in this particular scenario. They don't really have, to me, they don't have the right reporters to go in and ask those questions. Because they can't disentangle like the influence of quorum sensing, especially localized quorum sensing events, right, on this particular phenomena. Because even with their quorum sensing mutants, they don't look exactly the same, right? And if like you're going to relate that quorum sensing data to this one, to this final figure, then like in some ways they may have even seen differences between those two things, right? And again, biology is about balance, right? So like of course there's going to be stronger and weaker effects all like contributed from different systems. What I like about this particular paper, I guess, is that it is putting the emphasis, and I think it does a really good job of doing it, putting emphasis on these physical parameters, right? That like some of this complexity can just be explained through really simple things that are going on, and you don't have to dive into all this regulatory complexity. However, right, I would say regulatory complexity is still a big piece of this, right? And they're not able to successfully eliminate that from their experimental design. And I think that to me makes it that definitely weakens this last figure a lot, right? Because if they could show that this phenomena is purely based on some sort of like like the mechanical properties, right, of what's going on, then I think it would be a lot stronger. But like, I just know that it isn't, right? Like they haven't removed all those other variables.
Yeah, I mean, I feel like I'm, I feel like they're going around this direction, like looking at it from antibiotic treatments from a practical perspective. It's not going to be very useful because you're trying to hit the bacteria at a phase that's very, very early on in infection. So from a clinical perspective, I don't think it's going to be very useful at all because you're basically trying to make sure you get the bacteria at the point before they even cause an infection, the point where they're trying to settle in a place. But
Yeah.
that being said, there are plenty of other useful things for that understanding how bacteria behave in these. So like the way that they colonize certain areas. So I mean, biofilms are a big part of our gut. So how they
Mm-hmm.
recolonize the gut, how the, or even like as we in the very first like slide that we showed, all those different
Yeah.
things
Teeth,
could be.
pipes, bioreactors, right? It happens in ship hulls, like all these places, right? Like there are some sort of dynamics. If we could generalize some of those dynamics, that would be really cool, right? Because that would let us make better predictions, right? And then so as we're developing technologies, we can go down those paths. Yeah, human
Yeah.
health, not really sure what the interplay is. It could be there. This figure six
Well,
isn't doing it for me.
yeah, I mean, in that case, I'd have said like, okay, you've got like intravenous shunt catheters. They're quite an important place where you want to prevent biofilms. So you want to make sure that there's a lot of surface like engineering that goes into trying to prevent biofilms.
Mm.
That's gonna be quite interesting. I mean, I remember there's read this research about like shark scales and how the way they're designed prevents biofilms from forming on the surfaces of sharks, helping the
Nice.
hydrodynamics of those sharks. So stuff like that, like understand. So I mean, I feel like this kind of thing can give us more insights into like those initial physics of like how bacteria interact with surfaces and how those uses those surfaces to build biofilms and what we can do to potentially disrupt those interactions. Um,
Yeah. I think what I like is like I like the experimental model as this springboard, right? Like again, I really like the agar block and like the fact that they can see the wrinkles so close to the 90-degree agar angle. Um, like that is like to me that's a signal, right? Like it is something. We don't know how it's propagating through the whole regulatory network of the B subtilis, but it is, and I would like to know more about that. That's the most
yeah,
exciting thing for me. I think that's come out of this paper. And then knowing also that like you can predict some of this behavior just by thinking about bacteria as if they were self-propelled motes of different shapes.
yes, we can predict a cow of spherical shape and uniform density, and it will
Hmm.
tell us a lot about the. I mean, for bacteria, some of them are literally spherical and have a uniform density. So actually, there's a lot the mathematical models can do for predicting bacteria. They are the
Well,
simplest form of
but
life.
if we go back to if you think about that prediction that they used, right? That prediction it was based on 2D rods,
Mm.
right? Presumably that means that with more work in the theoretical space, they could generalize 2D rods into 2D, like what about those bifido shapes, right?
Oh,
Or like dipteroid shapes, right? Why are bacteria those shapes? Do they give them favorable qualities in certain physical scenarios? Right, and I think that like to me that's the interest that the value I see of linking it to the physics is because that then the field of physics becomes a way of generating hypotheses onto the natural world, right? Because you can say, oh, we predicted Mips would be different, right, when you change up the shape. Oh, here's another bacteria that is a different shape, right, that has similar biofilm characteristics. What would it do in this scenario? Does does the generalization still apply?
Yeah, I agree, and I feel like yeah, the physics are quite interesting. I feel like there could have been a lot more done in like statistically to validate the model that I don't think feel was done, and I feel like that would strengthen my kind of belief in this paper. But um, that's something
No,
that
but as a quick back, I know that it is discouraging your belief in the paper, but like when you look at it, I
hmm,
guess maybe that's the thing. Statistics is supposed to cut through that idea of like your eyes showed you the reality, but
yeah,
sometimes statistics are just right, like the eye, right, is just like a rough approximation of that, right? Like you can still see effect size and not like you can't see statistical significance, but you can see effect size.
yeah, that's true. But I feel like I can be tricked very easily into seeing effect sizes though, where because of just the way that humans perform patterns. Uh, But I
For sure.
do again. I feel like it. I'm convinced that this might be an effect that's happening. I feel like they could do more to expand on that model and make it more predictive. And I feel like
Mhm.
because yeah, I feel like the model fitting wouldn't degrade the paper. I feel like it would add a new thing, just a new
Yes.
way of understanding that could be built upon.
Yeah. And I agree. I don't think it would detract from the paper. I think that it would definitely enhance like that
Yeah,
particular part where they overlaid their points onto the predictions.
yeah, because again, though, I mean, when they did the experiments a couple of times, you see that the points are arriving in different. So the UV experiments, the points end up quite a lot lower than in the antibiotic experiments, and be quite interesting to understand and pass those effects because that. I mean, there are definite differences in the way the bacteria are behaving in those that the researchers hone in on, and I feel like that we could like bring in quite a lot more information. And it is just amazing that a lot of this is done with microscopy. There's very little like kind of there's no RNA. They don't like sequence like each of these islands to figure out what's the RNA stuff doing and when these bacteria are behaving this way. Um,
Yeah. Yeah.
so
Yeah, no, I mean, I think that that's where it's like that's the really powerful thing about doing biophysics in this way is like it potentially can erase some of those complicating factors and give us like yeah, give us an underlying principle that like we don't have to dive into all that complexity. But of course there will be tons of scenarios where you have to right understand the gene regulation a little bit better.
yeah, and there are lots of things I liked about this paper, like those images of striations. The way I mean, I wish they did talk a bit more about their image processing, like in their work, just like so that it'd be much easier for people like us to go off grow a plate of bacteria and then use the same analyses on different species of bacteria
Ah, yeah.
and see what patterns we see. I
Yes.
feel
Yes.
like that would be really cool. I mean,
Yeah,
we'd
I
have
think
to use
what
different
I
models,
mean, this
but
is just gonna. I think we've said a little bit about this. eLife is a very interesting journal, right? Like I saw that in
hmm,
the methods section, there's a place where you can like request more detailed protocols, right? You can
yeah,
like, send that email and say, I want to know more about this, right? And then that's a way to build like expertise amongst groups of people, right? Like, oh, now there's a group interested in this, so like maybe that's a collaboration that will form, right? Maybe that's a new paper that comes out, right? As things mix like that.
yeah. So yeah, I really like this paper. I think that it was very entertaining to read. I love the way that eLife set up their papers. Um,
Mhm. And yeah, I think that's great. That's all I want to say, I think, about it. How about you? You're good?
yeah, I am good. I'm just going to pull up. Yeah. So
So you gotta join us next week for our news week, where we will survey some papers to find something to cover in the following week.
yeah, and we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. I mean, there was a lot of biophysics in this paper, and today my brain's been a bit foggy because I just had my AstraZeneca vaccine, so we'll make of that what it is. But yeah, science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter with that hashtag #MicroTWJC. We both believe that peer review is this process where we can all participate in, and it happens even after the papers are published. So we hope that you've enjoyed listening to about microbiology today. And if you think you have something to add or found something unclear, please let us know.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. We'll see you then.